this thing has gotten much bigger, and that sterile jacket of cells surrounding the egg, after the zygote forms, the sporophyte forms, it gets stretched and stretched and stretched, and ultimately ruptures. Right? And then outside of that is what we call a perianth. Those are other leaves that surround that. So I can ask you, I can point to you that and say, what's the ploidy level? It's haploid. And then what's the term calyptra? What does it develop from? The neck and the venter of the archegonium. We'll see other examples of that. Thallus. All right, this one's upside down too. There's scales and rhizoids. The scales are multicellular. The rhizoids are unicellular. In most cases, they're coming at you through the plane of the section, so you just see a little circle that's cut in cross-section. Rhizoids are single cell, scales are multicellular. Here are parenchyma cells mostly storing water and not photosynthesizing. On the top surface, there are photosynthetic cells. On top of that are cells that have a heavy cuticle, and then there are pores that allow for gas exchange. Make sure you see the conocephalum in back, because the pores are really obvious on that. Okay, so you have on the top, you have that going on there. Pores are not homologous to stomates, though. They're analogous, they provide the same function. They facilitate gas exchange. But since this is a gametophyte, and stomates occur on sporophytes, one's diploid, one's haploid, they can't be the same thing. They're merely analogous. Okay. Naive capsule. All right, so this is the true moss. You don't see the foot, but it's pointed. The seta is elongate. You have a columella in the middle. That's a sterile column of cells. That's the capsule of the sporophyte. And then up here is an operculum that pops off. So it's like a cap. It pops off. In this case, these guys do not have eliders. Instead, they have peristome teeth. Peristome means around the mouth. So around the mouth, that is the top, this thing opens up. There are these teeth, and they respond to hygroscopic movements, changes in humidity. If you get them out of the lab at a light source with a scope, you'll see how they actually do this. They'll do like that. Right? And they're actually digging in and digging out the spores and ejecting them as the humidity changes. So you want to know for each of these groups, what is the mechanism that facilitates spore dispersal? We have eliders, now we have peristome teeth. It's going to get more exciting when we do the leafy liverworts and the sphagnum. Okay, so here's a leaf, single cell layer thick. So leaves, again, this is leaves of gametophytes, so analogous, not homologous to leaves of sporophytes. Difference is single cell layer thick, and there is a midrib. So in mosses, there is a midrib. The leaves are spirally arranged, and we'll learn how to distinguish those from leafy liverworts. All right, so this one I don't like. I mean, if you looked at this one, it's kind of like not much going on. You just see the capsule wall, and then you see these will be spore tissues that hasn't developed yet, and this is the columella. So I wouldn't sweat this one too much. That one's not very good either. I like that one. Okay, this one's good. It turns out in some of these mosses, in Niam anyhow, you have two separate plants. So they have these upright little plants sticking up. And at the top, they're going to make their gametology. In this case, these are antheridia, because you see a bunch of sperm cells in there. They're not round, but they're kind of globose. And then they're at the top. And then there's all these extra leaves in here. They are called paraphyses. And they are there to make sure that this stays wet. So they provide capillarity. The same thing happens. These things will open up a little bit more. The rain will fall, splash onto these, rupture these. And then these guys have to swim over all of these separate leaves. Okay, you think that's hard on the sperm, but it's better that there's moisture there. If these guys weren't here, the thing would be completely dry too quickly and they wouldn't get a chance to get out of there. So they provide capillarity. They conduct moisture and provide an exterior film of water to facilitate the sperm to swim over to the egg on a separate plant. Okay, now we got a movie. We'll see if this works. Get your popcorn back there? Oh, you want that one. I'm ready for my close-up, Mr. DeVille. All right. All right, here's the point. It's hard for me to show you everything you need to in one slide. These things are big. So we made this so you can see more of it, right? So as you're looking at slides, you don't have to make a movie, but do move the slide around so you know what's going on there. All right, this is one of my favorites. This is called moss protonema. So a spore is released from a true moss. It lands somewhere that's good for moss to grow, kind of moist. It's a haploid cell. Spores are always haploid. They divide mitotically, and they make a structure that's called a protonema. And then later, after this gets of a certain size, it'll make the mature gametophyte. So it's an early stage of development of the gametophyte. It doesn't look like the gametophyte yet. What it looks like is a branched filamentous green alga. That is exciting. Okay, here's 3 words to explain this. First word, ontogeny, that means development. Second word, recapitulate, which is English, a fancy word for repeats or just recap. Third word, phylogeny, is evolution. Ontogeny recapitulates phylogeny. So as things develop, the early stages of development are more likely to resemble the ancestral form from which an organism evolved. Okay, when I say that to you, you think about back in the good old days when you were just a small fetus in your mother's womb, and you're going to start reaching over here and seeing if you still have what? Gill slits. All right, so when you're a 6-week-old fetus, you look like a fish. Why are we looking like fish? Is that a sensitive topic? We share a common ancestor, right? So the rule is it's harder to change those early stages of development because they have more effect on the later stages. So they tend to retain the ancestral state. And then of course, I don't know, maybe you went to high school with people that still had gill slits, or there's the creature from the Black Lagoon, or I don't know, but you know what happened to them? They turn into the bones in your ear. That's what happens to your gill slits. Okay. So ontogeny recapitulates phylogeny. I don't have one, but for sphagnum peat moss, its protonema looks like Coleochaete. Okay? So we may be hiring someone for next year that does Evo-Devo, which is evolutionary development, which gets into that kind of stuff. Okay, Plintricum, I'm going faster now. Plintricum. This is a cross-section of the leaf. Looks like a spaceship, but don't freak out. This is the underside of the leaf, has abundant cuticle, and then these are like beaded curtains. So there's air spaces between those. There's single files of cells, and they're little files that go all the way in and out of the plane of the section along the entire length of the leaf. They provide capillarity. What are they called? Lamellae, L-A-M-E-L-L-A-E, lamellae. In some mosses, there's more cuticle, in some there's less. If there's less cuticle, they absorb water directly from the environment. If they have cuticle, they're more likely to move the water internally through hydroids and leptoids. Okay. Perella, now we're moving on to leafy liver. It's the last one I think I'm doing, except sphagnum. I got a couple of those. Cross-section through these, so you're seeing these are the leaves. There's leaves that surround all of these structures in the leafy liverworts. And they're around the male, we call them andrecium. They're just leaves that cover these antheridia, and it's called gynoecium if you're looking at the female with the archegonia. They provide capillarity, they keep these guys moist, facilitate the sperm motility by swimming through a continuous film of water. These guys will rupture, sperm will be released. They'll swim over to another branch. Here, so doesn't that look like emeralds, right? So here's a whole map, the whole thing you're looking at. You're looking right through it. It's hard to see, but here are these little pointy structures. Those are archegonia. And these leaves are the gynoecium. They're covering the tip of this branch, and that is keeping it moist and facilitating the sperm swimming in there. So when you're looking at a whole mound, focus up and down. As you focus through this leaf, you'll see the archegonia come into focus there. Okay, the other thing, you have to know these guys vegetatively. So how do we tell the leafy liverworts from the mosses? The mosses have spirally arranged leaves, sticking out in all directions from the stem. They almost always have a midrib as well. Leafy liverworts never have a midrib, and the leaves are sticking out in 2 directions. Okay, but it's a little busy here because this is really just a lobe of this leaf. So the leaves stick out in 2 directions, but it's like they have a thumb or a lobe. So they're doing this. And then there's a third set of leaf, that's this one here. That's perpendicular to the other 2, and it's on the underside of the stem that's laying on a rock or something that's moist. Those are called amphigastria. What does gaster mean in Greek? Stomach. Gastric indigestion, right? Yeah. So gaster, remember that for the belly. Why would a plant make a leaf on the underside of the plant away from the sun? Stupid plant. It's for water. It's all about water in the bryophytes. So it's going to help absorb water from the substrate and then move it along the plant. Okay, so know these guys vegetatively sporophyte. Are you guys ready for this one? Let's see if this one. Oh yeah. Here we go. All right, so here's the elders. They look like candy canes. There's the spores. They look like Christmas ornaments. So the mature sporophyte of Corella looks just like Christmas. Okay? Here's the jacket. Here's the calyptra. That's bearing it. This thing will split open when the calyptra rupture. It dries out. The elders twist, and the spores are dispersed. Okay. And then here's the rest of it. So here's a seta, and there's a foot, and then here's the rest of the gametophyte. That'd be the rest of the branch that it developed from. So a rounded foot in these guys. All right, last couple things here. This is a sphagnum leaf. Note this reticulate pattern. It's got all these diamond cells. Most of the cells in sphagnum, the peat moss, this is a separate, this is not a true moss, right? So we'll call it the peat mosses. Most of those cells are dead. When you see sphagnum, it looks yellow-green, right? There are living cells, small cells that are around these diagonal cells. They are green and photosynthetic, but most of them are clear and empty. So sphagnum is the only moss that grows in full sunlight. Why? Because it needs full sunlight, because it doesn't have enough chlorophyll-containing cells to photosynthesize. The cells are dead and empty, but they have a function. Their function is to hold water. There's holes in the walls, and they fill up with water, and this thing can hold 20 times its weight in water. In addition, the living cells make protons, they make acid, and that changes the environment it grows into an acidic environment, which favors the sphagnum and also the pitcher plants and things like that. So if you're growing pitcher plants, you can grow them if you can grow sphagnum, because they're going to make the environment acidic. That's going to slow decomposition, and then insectivorous plants can't get nutrients. What do? They catch bugs out of the air. Right? Yeah.
The non-photosynthetic ones, the hyaline?
Hyaline cells are the non-photosynthetic. So since they're acidic and they hold moisture, this was used by the Native Americans for diapers or to pack bandages in World War I. It's a good antiseptic dressing. Peat moss. Okay. And this is the last one of this right here. Yeah, that's just a whole just entertaining, pretty to look at. Don't buy peat moss because what they're doing is they're draining peat bogs and then dredging it out of Canada and then selling it to you. But if they drain the peat bogs, they remove the water, which makes it non-suitable for sphagnum, which means that then you get decomposition, and then you're into a positive feedback loop. You get global warming, and we all want to move north. Of course, we're all doomed anyhow, so there's that too. Okay, enjoy your freedom.